Um, this morning, I'm going to talk about different types of trials and some things to keep in mind, and then I'll hand it back over to Dr. Bangdiwala to keep going. So you saw this slide in the last session briefly, and this is discussing types of efficacy trials, specifically drug trials. And this is probably what most people think about when they think about clinical trials, are the drug trials. And so these are categorized by phase. The first phase is preclinical. So this is experimenting in animals. The population is animals. We're not testing in humans yet. We're basically just checking to see what the effects are in a living creature. And then we move on to trials in humans. And here we start with phase 1. This is the first time we're testing in humans. It's just going to be a small sample size, just a few healthy participants. So these are people who are not afflicted with the disease of interest. These are healthy participants who are volunteering. And we are specifically assessing preliminary data on safety and tolerability. So how well is this drug tolerated in humans? If the drug does well in phase 1, it can move on to phase 2, which is the first testing in patients who are afflicted with the disease. So these studies are typically anywhere between 150 participants to 350 participants, give or take. And primarily we're looking at safety and any adverse events going on with these participants when they take this drug. And we're looking at dosage. We're trying to find what is the right dosage for that's tolerable in patients with with the disease of interest. If the drug does well in phase 2, then we move on to phase 3. And this is typically when people think of drug trials, they're thinking of phase 3 because these trials are where the drug is compared to existing treatments, and it's going to be approved for use in the wider population. So these are usually a lot larger, 250 up to 4,000 or more. And these are, again, participants with the disease. Here we're looking at short-term efficacy. How much better does the new drug do compared to the control arm, which is maybe placebo or standard of care, depending on how that particular disease is treated. And we're looking at short-term safety. Then once a drug gets through phase 3, it's approved for use and people start using it, there's phase 4 trials. That's post-approval. And these studies are still looking at people with the disease who are using this drug after approval. And here we can actually look at long-term effects and continuing assessment of safety. So each of these has their purpose. And I just want to highlight that at every step along the way, we are looking at safety. Because remember, as Dr. Bangdiwala said in the last session, with our clinical trials, we are always considering our ethics. We are conducting experiments on humans, and it is of utmost importance to always be checking safety. Now I'd like to highlight the differences between infectious diseases and chronic diseases in clinical trials. And part of the reason this is part of my slides is because I work in infectious diseases. Most of the studies that I work on are dealing with a population of participants who either have cryptococcal meningitis or are prone to cryptococcal meningitis. And so my trials are different than a lot of trials for chronic diseases. So in infectious diseases, as I'm sure you all are all aware, of the differences the way these are affect humans. Just the infectious diseases are more acute, chronic diseases are chronic. And so because they're so different, then their trials are designed differently as well. Primary endpoints in an infectious disease trial, it's maybe in a prevention trial, we're looking at incidence of disease. We're maybe looking at short-term mortality because of the way a disease might operate. In chronic diseases, we're looking at symptom management, right? This is a chronic disease that we're dealing with. So we're looking at symptom management or quality of life. A similarity is possibly both of these types of diseases one of the endpoints could be absence of disease. Are we depending on the drug that we're looking at, or the intervention that we're looking at, are we able to actually cure? So that's a similarity. Infectious disease trials are often have sample sizes the several thousand. They don't have to, depends on what, what your primary endpoint is and what kind of power you're expecting to have. Chronic diseases can be large or they can be small. A lot of cancer trials are often very small. And some of that is because of the cancer only affecting a certain number of people. Timeline is probably the biggest difference between these two. Because of the primary endpoints that you're measuring in an infectious disease, you're maybe powering through that study in a couple of months, maybe a year or two. Whereas clinical trials for chronic diseases can be several years. I know at the beginning of the last session, Dr. Bangdiwala mentioned that, trials take time. How many are you going to do in your life and career as a clinical trialist? If we're focusing on chronic diseases, it may be several years before one trial is finished. And in infectious diseases, maybe it's just a couple months and then you're moving on to the next one. And the interventions are often very different, right? In infectious diseases, you can be looking at vaccines and medications. In chronic diseases, you can also be looking at medications. That's a similarity. But with infectious diseases, because of the nature of these diseases, right, that they are passed between people, a lot of times an intervention might be a public health measure where we're sampling entire communities and we're going to apply the intervention to an entire community to see if that can help with the transmission of the disease. Chronic diseases, that's typically just something that's affecting individuals. And so we're not going to necessarily have a public health intervention, but maybe we have a lifestyle management intervention. So that's some differences. And so, like I said, I work in infectious diseases. And this is a slide with too many words, but I just want to highlight that I said I work in cryptococcal meningitis, which you likely know is a fungal infection that causes meningitis. It's 100% fatal without antifungal therapy. And most of those deaths from meningitis occur between 3 to 18 weeks after diagnosis. And so what I've listed here are 3 of our trials that we've done over the last 10 years. The ENACT trial, the AMPLEX trial, and the ASTRO trial. Each of these looked at a different antifungal therapy or a therapy that was shown to have antifungal properties, like the ASTRO trial looked at sertraline. And you can see that the primary endpoint for each of these trials was either measured at 2 weeks or 18 weeks, super fast. And then all these secondary endpoints were also, 18-week survival, neurocognitive abilities, depression, things that are measured very quickly. And so each of these trials, we were able to measure that primary endpoint at 18 weeks. And then we, of course, followed surviving participants through a year just to collect some year-long data and to provide care for those participants through 1 year. Okay, so this slide you also saw in the last session. Dr. Bangdiwala discussed pragmatic versus explanatory trials. And I thought I would just highlight this example that was displayed. This is back in 2020, in March. All of our cryptococcal meningitis trials were halted, like most trials around the world, and everybody was focusing on COVID. And so it was happening in real time. It was happening everywhere. An efficacy or explanatory trial was not going to be useful. All right, we couldn't control all of the circumstances. We had to be pragmatic. We had to deal with how things were in the real world. And so what we did, my principal investigator was aware of hydroxychloroquine as having been potentially useful for SARS decades earlier. And so we thought we'd try it for SARS-CoV-2. This was before any political issues and considerations of hydroxychloroquine. We all hoped it would be something useful. So we conducted an internet-based trial. We literally launched in REDCap. We promoted the trial as best we could. People were given a link to our website where they could access the survey. that they filled out themselves, and depending on what they submitted through their survey, they were eligible or not. We randomized them to either receive hydroxychloroquine or a placebo. We shipped it to them. They responded to survey questions, and that's how we got our follow-up data. It was very real world. And because of the way we were able to, all of the resources were going towards COVID at the time, we were able to implement that very quickly and get the answer very quickly that no, hydroxychloroquine was not actually helpful for post-exposure prophylaxis and preventing COVID-19. So that's an example of a pragmatic trial. This slide is describing, it's the Precis wheel, which was developed to help trialists designing clinical trials to consider where they would like their trial to be on the pragmatic explanatory continuum, because it's not one or the other always. There's different elements to consider: eligibility, recruitment, setting, organization, flexibility of delivery, flexibility of adherence, follow-up, the primary outcome, and primary analysis. And each of these elements could be explanatory or pragmatic. In section A, we're looking at a Precis summary of a randomized controlled trial of self-supervised and directly observed treatment of tuberculosis, where everything is rather explanatory. And in section B, we're seeing a Precis summary of the North American symptomatic carotid. Oh, I'm going to say the NASCEC is how I'm going to say that. And looking at high-grade carotid stenosis. And so this is much more controlled. And then you can see that this tool is useful for helping you understand, okay, this element, how should the intervention be delivered? I can be a little bit more pragmatic here. Or how is the primary outcome? How is it relevant to participants? Maybe I need to be a little bit more controlling here. Okay, moving on, different types of trials. Pilot Vanguard trials are also known as feasibility studies. These are very important, especially when there's like a new type of intervention that's never been done. It's not necessarily just oh, we've got a drug and we can deliver it, we can randomize and give the drug or the placebo. If there's a new type of intervention that is going to need some thought taken to it. I'll give you an example. We have a trial going right now for cryptococcal meningitis, and we have a colleague who wants to implement a keto diet. So it's possible that somebody on a keto diet has if they take an antifungal therapy, that the therapy is going to work better for them. But our trials are conducted in Uganda, and they have a very specific diet there. And so trying to implement a keto diet on top of that, on top of the trial, might be impossible. And we are reminding our colleague that they have to conduct a study deciding the feasibility before we can implement a large, expensive, full-scale trial. I'll also highlight the reasons for piloting. So the process. This is assessing the feasibility of the processes that are key to the success of the main study. Resources, we want to make sure that we're assessing the time and resource problems that can occur in the main study. We want to test this out on a smaller scale. We want to look at just a few elements before we're going to a major, large, expensive, full scale. Management. Do we have the humans ability to manage the humans? Do we have the staff for this? Do we have the ability to call patients back? Do we have the ability to manage patients? Do we have the data management abilities? Do we have the staff to manage data, analyze the data? And scientific, do we have the ability to assess the treatment, safety, dose, response, etc.? So conducting a pilot prior to the main study, it can enhance the likelihood of the success of the main study and potentially help avoid doomed main studies, right? So the more you test ahead of time, you're testing whether you can implement the things on the ground and maybe like a multi-center study. You're making sure you have the personnel. When you have all that set up in a pilot study, you're setting yourself up for success in the main study. A pilot study should be well designed with clear feasibility objectives so that you can say at the end of the study, yes, this is feasible, yes or no. It should have a clear analytic plan. Uh, it should be used cautiously for determining treatment effects. It should, maybe you need it for determining an effect size, but it should be cautiously used for that. And ultimately, it should be scrutinized the same way that a full-scale study is, and the results should be published. Other people should know about how you were able to set yourself up for success in the main study because it can help them to set themselves up. They can learn from what you did. Actually, I'll go back to our hydroxychloroquine trial. After we implemented that internet-based pragmatic trial, I think we've also published, on top of the main study, we published at least 2 more papers on lessons learned through that implementing that rapid-faced internet-based trial. I published one on tips and tricks for survey studies in REDCap, and another one of my colleagues did lessons learned for patient care via internet-based trials. Okay, now moving on to platform, umbrella, and basket trials. So all of these 3, platform, umbrella, and basket, can be thought of as like a master protocol. So I don't remember if it's been discussed yet during these sessions, but a protocol is basically the giant overarching document that details every aspect of the trial, from the reason you're doing it to most of the details about how you're going to do it. And this master protocol, in some cases, could involve multiple therapies, or it could be dealing with multiple diseases with a single overarching protocol. So most trials are just one protocol dealing with one disease and one possible intervention. And sometimes, to manage resources, it makes sense to try and handle multiple drugs or multiple diseases at once. Some of the advantages include, like I said, operational efficacy, or sorry, operational efficiency. So that's like the same infrastructure is developed and implemented for multiple sub-studies. Like you're using the same infrastructure for site selection, patient screening, data management, etc. You can also save patient resources and make the trial more appealing to participants. So sometimes, if there's multiple interventions, multiple therapies offered within one protocol, a participant might have more of a chance of getting being randomized to an experimental arm, right? And so they're like, okay, I don't have as much of a chance of getting placebo. I want to be involved in this trial. Another advantage is information borrowing across sub-studies. So it could be made available, and such trials could have innovative statistical methods because you're dealing with so much at once. So to highlight the difference between all of these different types of master protocols, there's a basket trial, which is designed to evaluate a single investigational drug in different disease populations. So maybe there's a drug that has proved to seem effective in different types of diseases. If that's all contained in one protocol, that's a basket trial. An umbrella trial is the inverse. It's designed to evaluate multiple investigational drugs in a single disease population. And then a platform trial is kind of designed with features of both, or it's sort of takes the nature of the basket trial or the umbrella trial and makes it perpetual. So what that means is, like, an umbrella trial might have a master protocol that says, okay, this is our set of sites, this is our set of drugs, this is our disease population, and we are going to enroll this number of participants and randomize them to different arms, and when it's done, and we will analyze. A platform trial could take that and say, okay, while we are randomizing to this set of drugs, another drug might be available, and then we'll start randomizing to that. And Once we have enough participants in one drug, we'll stop and analyze that, but we'll continue enrolling for the next drug and continue on. So to continue this thought, I'll take us back to this horrible slide that I made that has way too many words again, and remind you that I told you about 3 different drugs that were all looking at cryptococcal meningitis. They had different antifungal therapies, mostly the same endpoints. And so now you can see that these were done in 2015, 2019, 2020. Now in 2025, my team has been able to take everything we've learned from that, and we've started a platform trial for all of these antifungal therapies to use our same infrastructure. So over the last 10 years, we have built our infrastructure at our sites in Uganda. We have a great team. We have our site set up. We have a system. And now we can actually implement a platform trial where anytime there's a new potential antifungal therapy, we can roll it into this like umbrella perpetual platform trial. So we're calling it the Platform CM trial. It's an open label randomized trial with potentially multiple interventional arms to compare efficacy and safety. One thing I'll highlight with the platform trial is that it'll have platform eligibility criteria. So whether a person is eligible to participate, they'll have to meet the eligibility criteria for the platform in general. But then each of these antifungal therapies will have its own eligibility criteria. And so in that way, suppose at a certain point in time, say tomorrow, we have a participant who shows up and we have 2 experimental antifungal therapies going at this time. We just don't have the 3rd or the 4th yet, right? Those are still they're still in phase 2. This is a phase 3 and we have 2 therapies. In that case, we have actually 3 arms. We've got antifungal therapy 1, antifungal therapy 2, and the standard of care. And so if this participant is eligible for both of those interventions, then they could be randomized to any one of those 3 arms. And when each of those antifungal therapies reaches the point of enrollment where they're able to be analyzed, the analysis set for each drug includes people who are eligible for that drug at enrollment. And so the 2 drugs get to share controls, and that's saving us resources and really helping with the trial. This trial is designed to be perpetual, to continue as long as we've got new therapies coming in. Another benefit of that is if we don't have any therapies going, we won't be able to randomize, but if we have at least one therapy going, we're able to randomize and provide care to participants. Because we work in Uganda, which has limited resources. And so if there's not a trial going on, these patients in these public hospitals are not able to access any care whatsoever. So by designing a platform trial, we have a system set up to continually study different potential antifungal therapies and therefore continually provide care to these participants. Like I said, this is a master protocol. So all of these drugs are going to share the same protocol. They have the same endpoint, which is that 18-week survival. They share the same sites, the same trial staff. What's really nice is that they share the same DSMB, which I don't know if that's going to be touched on in this session, in this workshop, but getting a DSMB, a data safety monitoring board together, where everybody agrees and you have somebody of every expertise that you need, can be difficult to get a group of colleagues together to be on a DSMB. So to have one set and be able to use that for multiple therapies, multiple drug trials within one, is also saving everybody resources. So that is the end of my section of describing all of these trials that I'm familiar with. And now I will thank you and pass it back.